hippocampus. So if you look at just neural activity in the hippocampus as an animal does some task, in our case, we're just going to have head-fixed mice that are running down a 1D linear virtual reality corridor. What we'll see is that the activity will kind of tile out the space really nicely. This is going to be in CA1, CA3 of the hippocampus. One of the intriguing things about this activity, it's this kind of sparse rotating vector through the population, is that if you now change the environment, change the task, in our case we just changed the virtual reality environment that the mouse was running through, you're going to get a brand new sequence of activity, right? And this sequence, this is called remapping, and this sequence is in the exact same cells, right? So somehow this network, the same network, can support a sequential kind of activity pattern, and also be able to instantaneously remap or adjust itself to take different inputs and kind of tile it out into a completely different rotation. So we're interested in like what kind of mechanisms, what kind of circuit will allow you to do that. We're going to start by looking at CA3. CA3 is really the area that starts these kind of mapping and remapping processes in the mouse. And the way we're going to start looking at the dynamics is we're going to just start zapping different cells. We're going to use two-photon optogenetics. We're going to stimulate them for 10 milliseconds briefly, just 3 cells in this kind of sparse ops and prep. And we're going to look at the interactions between these cells, how much they're pushing and pulling against each other. One way to think about this is if we stimulate cells together versus by themselves, what's the delta, what's the difference, what's the delta, and that might be a sense of the interaction. We're going to push that a little bit further. We're actually not just going to look at sets of cells versus by themselves. We're also going to play in sequences. We're going to look at millisecond timescale interactions, which means just stimulating cells in 1 to 3, 1, 2, 3, etc. We're going to explore millisecond sequences up to 100 milliseconds, and then we're going to explore sequences of up to 2 seconds. This is the kind of data we get. So we're going to have, the third cell in the sequence. We can see that, for example, in this particular one example, stimulation at 25 millisecond lag, this cell is kind of relatively excited. And then we're going to look at 100 millisecond lag. The same cell is going to be a bit weaker in its response. If we just plot out all of our lag conditions on the x-axis for this one cell, we're going to see exactly that. The cell is kind of disinhibited and then more inhibited at the 100 milliseconds. This gray area in the background, this is just if you stimulate the cell by itself, what it looks like. Okay, so we run this experiment many times, many mice. We can pull all the data together. What does it look like? What we see here is that there's this, if you stimulate cells simultaneously, you get a bit more excitatory drive than you would expect if you stimulate the cells by themselves. But then rapidly this turns into inhibition. So the 25, 50 millisecond lags, you get this inhibitory phase, and then it kind of evens out, and the longer time scales, you don't see anything. If you just look at the correlations of the repetitions with respect within a single lag condition, you'll see that the stimulation is relatively correlated at these short kind of millisecond time scale lags, and it's not significantly different from shuffle and these longer lags. All that's saying is if you're stimulating cells, with 2-second sequences or something like that, you may as well have stimulated them by themselves. There's no real memory in the network. It's really at these less than 100 millisecond timescales that the interactions are occurring within this network. This isn't the full picture, however. If we kind of expand this out and look at individual experiments that we performed, sometimes it does look something like the average, where this is just an example cell. It's excited, then inhibited, and then evens out. But sometimes we get the exact opposite effect. So here we have a cell that's inhibited more if you stimulate it simultaneously with a bunch of other cells, and then it becomes excited if you stimulate it with some sequence. One thing we do kind of, so the data is actually quite diverse, it's quite complex, but one thing we do kind of see across all of our examples, and you can kind of see it here, even though the data is noisy, is that nearby lags are more similar to each other than you expect by chance. So this kind of, especially this 100 millisecond time scale, whatever effect you have with respect to your other cells around you, it's smoothly changing over time, right? So we're kind of zoomed in at the time scale at which these interactions are changing, right? Okay. This kind of gives us an excuse to basically smooth out our data, so we can have fit these kind of nice smooth spines through the data. And again, these are kind of, to kind of just summarize this experiment, we're measuring the effective interactions between these cells. This is, of course, not monosynaptic. So, synapses don't change from excited to inhibitor or anything like that. These are just effective network interactions, the pushes and pulls between these cells over time. We're finding their EI balance. They dissipate after 100 milliseconds. They're kind of smoothly changing at these 25 milliseconds time scale. Now we can ask, okay, given these empirical observations, what does this mean for the CA3 network dynamics? And the approach we took is we actually just sampled these into an RNN, right? This is very not, our data is not very mechanistic, so our RNN, it does not have to be mechanistic either. We're really just describing pushes and pulls between cells, and that's what the RNN can do. The one difference, or the one change we do have to make to kind of classical RNN modeling is we have to add these lag-dependent interactions. And that's kind of pretty straightforward to add. You just say, like, for 100 milliseconds, you integrate, or I guess stepping back, you turn your weight matrix into a weight tensor. So your third dimension is just describing this kind of lag dimension that we have on the x-axis here. These are just 3 example weights, basically. You sample these into RNN, and then for each weight, you just integrate over the last 100 milliseconds of that cell's activity, of the presynaptic cell's activity, and you run it. So what do these add? What do these kind of empirical lag-dependent interactions add to the network? So we simulated giving, for example, nearby, 2 input vectors that are very nearby in state space to this network and seeing what it does with them. We can look at the trajectories. Immediately we see they're really funky looking. This is in the middle here. This is kind of this empirical lag-dependent RNN. You give it 2 similar inputs, and they're going to kind of spin around all the way down to 0. And this is very different from like a standard RNN, at least in our simulations, where they kind of rapidly converge just because there's some dominant dynamics in the network. What this leads to behaviorally for the RNN is that it can keep inputs separate for much longer. Even if you slow down like a standard RNN, such that the total amount of activity duration is the same as with our lag-dependent RNNs, the separation or just the distance between these trajectories converges much faster. Okay, so we have these stable networks that are keeping inputs separate. One way to think about what this can be useful for is we can kind of parameterize the input, basically take an input vector and just slowly drag it around state space and see how that changes the fixed points of the network. And when we do that, what we see is that there's this, when we say how does the fixed point change, we can just throw them into like PC1, 2, 3 and project them there. But what we can do is we can ask what the speed of that change is, right? How quickly are the dynamics changing as you slowly change the input? And what we see is we have periods of quiescence, and then we have these kind of little spikes. And these spikes represent times when the dynamics really shifted around, when you very like infinitesimally small change of the input. So this is cool. Can this do what we want to do with these networks, mapping and remapping? As a proof of principle, we can just simulate input to these networks. That's kind of biologically plausible. And the answer is yes. If you have some background grid cell activity and then you give kind of bursts of dentate gyrus-like, very sparse bursts of activity to these cells, you'll start these kind of chain reactions that form these sequences. And importantly, all you have to do to have a remapping event occur is you can just change the phase of this kind of background grid cell activity. And this is also kind of, at least experimentally, in neural activity, we do see this happening where around remapping events, grid cells don't stop being grid cells. It's not like all your connections change, but really the relative phase of your grid cell inputs changes. Okay, so this is kind of a proof of principle. This leads us to a working hypothesis where we can have different input patterns. They can be overlapping. There just need to be very minor differences. You can pass them through this dynamical reservoir. This is what we're kind of describing CA3 as, where it has very brief millisecond timescale dynamics that are nevertheless very good at extracting small differences in the input. And this leads to these kind of maps. How do we test this kind of working hypothesis? I'm just going to briefly describe a couple of approaches that we're starting to do. So, the first question we can ask is if the sequencing of place fields is occurring due to these millisecond timescale dynamics, then cells that are in a place field sequence should have stronger interactions between each other than just random cells in the network that might not even be firing. So the way we tested that was we performed a closed loop experiment. So we have the mouse running on this linear VR corridor. We quickly analyze that data, extract the place field locations, and then we go back and we stimulate those place fields all in sequence. Again, in a 25 millisecond lag sequence, so 100 milliseconds of stimulation. These are very brief pulses of stimulation. For the hippocampus people here, we're basically playing in replay events to the network. So we can do that, or we can stimulate, just pick 5 other cells, but stimulate them the same way in a sequence, but they're not related to each other in place fields. And we're kind of asking, are the interactions stronger? And we do find that that indeed the interactions between those cells is stronger is measured by how disinhibited or inhibited they are when they are in that sequence. Okay, so that kind of fits with our model. A second approach we took, and this one's a bit came out of another observation, which is what happens when we look at all the other cells in the network, right? Where we're playing in these very brief pulses of stimulation, can the network even tell what we're doing? So if we don't look at the cells we're stimulating, we look at the recruited population effectively, how sensitive it is to our stimulation, how sensitive to different types of stimulation. And one kind of striking observation we've made is that if you just either do t-tests on single cells or if you just train a linear decoder to tell apart what kind of stimulation you're doing, you completely fail. You're always at chance level. And this is this kind of black, this is a regular linear decoder over here. We're looking at our model, however, we can make a further prediction. We can say, well, what's really happening is as you're running through this corridor and the seconds time scales, the input is dragging you through state space, right? And in our model, what that means is the dynamics you're feeling are going to be very, very different. So all we have to do to actually be able to identify the recruitment patterns to do different stimulations is we should allow the linear decoder to rotate as the mouse is running through the corridor. And when we did that, just by bending the position basically and training different linear decoders, we were able to discriminate quite well, well above chance, what the stimulation patterns were. So that's the story so far. Just to summarize, causal interactions are very dense. They can be both facilitated or inhibitory. They can switch, varying over these tens of milliseconds timescales. RNN seeded with such interactions act as very good dynamical reservoirs, and interactions are stronger between cells that form place field sequences, and the network recruitment is very dependent on ongoing navigational activity. Thank you. I'm happy to take any questions.